Hi, I'm Kaylin, and I'm going to be presenting my work over the past year with Abigail and Dr. Agarwal in Apoptosis and Crithidia fasciculata. Just a brief overview of what I will cover. First, I will talk about the background information for the project, and then I will talk about the genes that I studied, and then I will talk about the CRISPR-Cas9 optimization process, and finally, Abby will talk about our drug selection experiments. First off, what is Crithidia? It is a unicellular eukaryotic parasite that is defined by its single specialized mitochondria called the kinetoplast. It infects mosquitoes and not humans. So it belongs to a group of pathogenic kinetoplastids. It is most closely related to Leishmania, which is spread by the sandfly and causes leishmaniasis. It is also very closely related to Trypanosoma brucei, which is spread by the tsetse fly and causes African sleeping sickness. It is also very closely related to Trypanosoma cruzi, which is spread by the triatomine bug and causes Chagas disease. All three of these diseases are classified as neglected tropical diseases by the World Health Organization, which means we need better treatments for them. Crithidia is a good model kinetoplastid because it is very easy to culture. It has not been shown to infect humans, which means it is safer to work with in the laboratory than its pathogenic relatives. It has a sequenced genome, which means we know exactly which portions of the genome we are characterizing in our work. And finally, it is very similar to Leishmania and Trypanosoma species, which means what we learn in Crithidia can likely be inferred to them and used to make better treatments for the diseases that they cause. So we specifically are studying apoptosis in Crithidia. The classic hallmarks have been observed, such as cell condensation, mitochondrial depolarization, and chromatin fragmentation. However, it is understudied. And that is because the pathways are very different than they are in mammalian cells, which is good. It means they are parasite specific and therefore will be good drug targets. Now, apoptosis in kinetoplastids serves a very different purpose. In the mammalian cells, it helps to maintain the integrity of the organism through protecting development and preventing cancer. However, in a unicellular organism, if the cell undergoes apoptosis, the entire organism dies, which may seem a little bit strange that an organism would want to do that. However, it is hypothesized that it is a form of selfish altruism that allows for selection of the fittest parasites and avoids hyperparasitism in the host, which allows it to evade the immune system for longer. Next, I will be talking about the genes I was interested in studying. The first gene is the BAX1 inhibitory protein gene, and here you can see it complexed with the BAX protein, and the structure was solved in E. coli. It is a hypothetical protein whose function is inferred based on gene ontology. It localizes to the mitochondria, and it is an inhibitor of apoptosis, and it does so by inhibiting the apoptosis promoting BAX protein. The other gene I was interested in is PGAM5, or phosphoglycerate mutase family member 5. It is also a hypothetical protein whose function was inferred using gene ontology. It also localizes to the mitochondria. However, this one is pro-apoptotic, and it does through activation of ASK1, which is a kinase. And here you can see a picture of the protein structure in Homo sapiens. Now, humans do have a PGAM5 gene. However, it is considerably different than the one that is present in kinetoplastids. Both of these genes are well conserved across all species of kinetoplastids and very similar. Therefore, what we learn in Crithidia is likely going to be able to be applied to Leishmania and Trypanosoma species. Next, I will talk about the CRISPR-Cas9 optimization process. So just a brief overview in case you are unfamiliar with CRISPR, what happens is guide RNA binds to the Cas9 complex, which guides it to the section of the genome where we want to initiate the double-stranded break, which is in the target gene. So there are two ways that it can repair this break because breaks are bad and cells don't like that. The first way is non-homologous end joining. What happens in that is base pairs are chewed off on either side and then replaced at random. This can initiate a mutation, or it can be repaired correctly, although that is more rare. The other method is homology-directed repair, and that is where the homologous chromosome is used as a template to correctly repair the damage. Now, Crithidia, like other kinetoplastids, is diploid, and it does prefer the homology-directed repair. So it does not tend to do non-homologous end joining, and we can use that to our advantage by inserting a homologous repair cassette that will be used in place of the homologous chromosome. And what we can do with that is insert a drug resistance gene in place of the target gene. So how do we generate the homologous repair cassette? First, we amplify 3 fragments, 2 homology arms that are 500 base pairs upstream, and then 1 500 base pairs downstream of the target gene, and then a drug resistance cassette. Then we use fusion PCR to put those pieces together. Finally, the fusion PCR product is cloned, and then once we have enough DNA generated, we can do transfection. So this is how the fusion PCR process works. The light blue strands are the homology arms that I mentioned previously. Those are found in the Crithidia genome on either side of the target gene. The green strand you see represents the drug resistance gene, which comes from a plasmid. The part where you see them overlapping in the upper part of the fragment and the upper part of the picture is where they are complementary, so they will bind during the annealing stage of the PCR, along with the nested primers, which are seen in orange, which will then amplify that entire long piece into one long strand of DNA, which is our homologous repair cassette. So over the past year, I've successfully generated the homologous repair cassette for the BAX1 inhibitory protein. Here is a picture of the gel electrophoresis of the PCR product. In the first lane to the right of the ladder, you see the drug resistance gene blastocidin alone. In the other 4 lanes that are higher, you see the blastocidin fusion product. And since it is larger, you can see that the 2 homology arms have been added on either side. This increase shows that we have successfully added those homology arms and created the homologous repair cassette. The PGAM5 fusion process is still being optimized. However, we have generated all the necessary fragments. In the first lane labeled BLAST, you see the blastocidin resistance gene. And then in the second one labeled PURO, you see the puromycin resistance gene. And on the one labeled 5', that is the 5' homology arm that is 500 base pairs. And then next to it is the 3' homology arm that is also 500 base pairs. The fusion process is still being optimized, but this shows that our primer design was correct and that they can correctly make the fragments. So fusion PCR just needs a little more work. And finally, Abby will be talking about our drug selection experiments.
Hello, my name is Abigail Della Penna, and today I will be talking about the drug selection portion of this experiment. In the drug selection protocol, we had 2 selection drugs that we chose. One was blastocidin and the other was puromycin. For each of our 2 selection drugs, blastocidin and puromycin, we had 2 replicates. Each replicate was inoculated with 1 milliliter of Crithidia. We then allowed the Crithidia to grow until it reached its logarithmic growth stage. Once it reached its logarithmic growth stage, we were then ready to add drugs. For our puromycin drug selection, we added 1 microgram per milliliter of puromycin to one of our treatment groups. And to our other treatment group, we added 50 micrograms per milliliters of puromycin. We also had a control group which had no drugs. After the addition of drugs, we counted Crithidia for the next 7 days using a hemocytometer. These are the results from our drug selection counts of Crithidia over 7 days, and this is all for puromycin. The blue line on this graph represents our control. This is our group with no addition of drugs. The orange line on this graph is our P1 group, or our 1 microgram per milliliter concentration of puromycin. And the gray line represents our P50, or our 50 micrograms per milliliters of puromycin. As you can see by comparing each of our treatment groups to the control, you can see our P50, or 50 micrograms per milliliters of puromycin, inhibited the growth of Crithidia, while our addition of 1 microgram per milliliter of puromycin led to an increased growth of Crithidia. We are not sure what led to this increased growth of Crithidia, but since this increased growth was seen in both of our replicates, we are assuming that since 1 microgram per milliliter is a very small concentration of drug, it is not enough to affect the growth of Crithidia. Based on the results from our counts of our Crithidia for our puromycin drug selection, we would proceed with using our P50 concentration, or 50 micrograms per milliliter of puromycin, because it led to an inhibition of growth of our Crithidia. We are still in the process of doing our blastocidin drug selection. Thank you for listening.